This is a DNA double helix which consists of two strands. This is the 5' to 3' strand, and this is the 3' to 5' strand. They are coiled, they are paired by means of hydrogen bonds. These are the parental strands or template strands. Just before replication, what happens? The two strands cannot be uncoiled and separated over the entire length of the DNA because it is energetically expensive process. The two strands uncoil, they are unzipped and they become free. They separate from each other and they become free from each other over a short distance, over a short length of the DNA, over a short stretch of DNA. In this portion, the replication process will start. This is the 5' to 3' template strand. This is the 3' to 5' template strand. They are free from each other. They are exposed so that on each template strand, a daughter strand can be synthesized. On the 3' to 5' template strand, a daughter strand is synthesized. On the 5' to 3' template strand, a daughter complementary DNA will be synthesized. So for this, the two strands must be free from each other. They must be uncoiled. They must be separated so that they can serve as template strands for the synthesis of new daughter DNA strands. So over a short distance, over a short length, the two DNA strands separate from each other. This portion where the two DNA strands separate, this part is called replication fork. This is the DNA double helix. The two strands are separated. This part is Y-shaped. Separation of the two DNA strands produces a Y-shaped structure which is called replication fork. This is a replication fork. Now how replication starts? The DNA double helix, the two template strands are separated from each other over a short distance. This is the 3' to 5' template, and this is the 5' to 3' template. The enzyme DNA polymerase binds to 3' to 5' template. The enzyme DNA polymerase lays down a daughter DNA strand in 5' to 3' direction continuously on this template. On 3' to 5' template, DNA polymerase synthesizes a new DNA strand which is complementary to the parental strand, and this new DNA strand is synthesized in 5' to 3' direction. Because DNA polymerase can synthesize DNA in one direction only, 5' to 3'. So this is the daughter strand which is formed continuously on 3' to 5' template by DNA polymerase. So this strand is called leading strand. This daughter strand is called leading strand, and the synthesis of this DNA is called continuous synthesis. Now this is the 5' to 3' template, but the DNA polymerase can synthesize DNA in 5' to 3' direction. So therefore, when the continuous synthesis is occurring, the leading strand is being formed. At the same time, DNA polymerase synthesizes DNA strand in the form of short segments on the 5 days to 3 days template. This is the 5 days to 3 days template. On this template also daughter DNA strand is formed, but this daughter DNA strand is formed in the form of small separate segments. On the 3 days to 5 days template, DNA is synthesized continuously, but on the 5 days to 3 days template, DNA is formed but in a discontinuous manner in the form of small fragments, and each fragment is synthesized in 5 days to 3 days direction, because DNA polymerase can synthesize DNA in 5 days to 3 days direction. So when DNA polymerase binds to 5 days to 3 days template, it synthesizes DNA in 5 days to 3 days direction, but in the form of small fragments. These are called Okazaki fragments, and each DNA fragment is laid down in 5 days to 3 days direction. 5 days, 3 days, 5 days, 3 days. Because DNA polymerase has the ability to synthesize DNA in one direction only, 5 days to 3 days. This is called discontinuous synthesis. And the Okazaki fragments are later on joined to form a DNA strand, which is called lagging strand. This is the leading strand, which is formed first and it is continuously synthesized. This is lagging strand because it is synthesized in a discontinuous manner on the 5 days to 3 days template. But remember, DNA is synthesized by the same enzyme, DNA polymerase, and in one direction only, 5 days to 3 days. 5 days to 3 days, 5 days to 3 days. This is also formed in 5 days to 3 days direction. But this is synthesized continuously. This is synthesized in a discontinuous manner. So this is the DNA strand, which is called leading strand, and this is the DNA strand, which is called lagging strand. So leading strand is formed on 3 days to 5 days template. Lagging strand is formed on 5 days to 3 days template by the same enzyme, DNA polymerase.